Hello everyone, myself Babita Suman and my topic is cells of immune system. Cells of the immune system. In this first we discuss about hematopoietic stem cells. All the blood cells arises from a type of cell called hematopoietic stem cells. Stem cells are cells that can differentiate into other cell type. They are self-renewing. Hematopoietic stem cells give rise to lymphoid and myeloid progenitors. All the lymphoid cells originate from lymphoid progenitor cells and all the myeloid lineage arises from myeloid progenitors. Hematopoietic stem cells is a multipotent stem cells, which means multi means many, potent means potency, means these cells have a capacity to differentiate it into different type of cells. So they can differentiate it into common myeloid progenitor cell, common lymphoid progenitor cell and erythroid progenitor cells. Myeloid progenitor cell can differentiate it into neutrophil, eosinophil, monocyte, mast cell, basophil, while monocyte further divided into dendritic and macrophages. While lymphoid progenitor cell can differentiate it into B cell progenitor, natural killer cell, T cell progenitor. T cell progenitor further divided into memory T cell, cytotoxic T cell, helper T cell, and B cell progenitor can divide it or differentiate it into memory B cell, plasma cell, while erythroid progenitor cell can divide or differentiate it into erythrocytes and platelets. The cells which involve in innate immunity are eosinophil, basophil, natural killer T cell, macrophage, dendritic cell, mast cell, neutrophil, and natural killer T cell, and their responses is rapid. While the cells which involve in adaptive immunity are CD4 T cell, CD8 T cell, B cell, natural killer T cell, and their responses is slow. The cells like natural killer T cell involved in both innate as well as adaptive immunity. Now we discuss the cell which arise from myeloid lineage. In this first we discuss about neutrophil. Neutrophil is a cell which constitute about 60 to 70% of total leukocyte. It is a principal phagocytic cell of innate immunity and stained with neutral dye, that is hematoxylin eosinate of methylene blue, and it is a 3 to 5 lobe structure. Neutrophil are produced by hematopoiesis in the bone marrow. Hematopoiesis is the process of formation of cellular components. Cytoplasmic and myeloperoxidase marker are the common marker that are used to identify human neutrophil. Movement of circulating neutrophil into tissue known as extravasation. Low count of neutrophil known as neutropenia. Granules which is present in neutrophil are proteases, protein defensin, antimicrobial, protease inhibitor, and histamine. Now we discuss function of neutrophil. Neutrophil are polymorphonuclear and phagocytic leukocyte that comprise the first line of host immune response against invading pathogen, and the function of neutrophil is phagocytosis, which means engulf the foreign particles, and also the ingestion and destruction of microorganism or other foreign particles. They are also important effector cell during tissue injury induced inflammation. Now we discuss about eosinophil. Eosinophil is a principal defender against parasite. It is a type of disease fighting white blood cells. Eosinophil is like neutrophil are motile phagocytic cell that can migrate from blood into the tissue space. It is stained with acidic dye, eosin, picric acid, and nigrosin. It is about 2 to 5% of total leukocyte WBC. It is a two-lobe structure and phagocytic cell, and the low count of eosinophil known as eosinopenia. Granule which is present in eosinophil are ribonuclease, cytokines, chemokines. Function of eosinophil are like these cells trap the substance and move towards the inflamed areas, and also participate in immediate allergic reaction, also modulate inflammatory response. Higher concentration is observed in endoparasitic infection. Now we discuss about basophil cells. Basophil cells are the cells which stain with basic dyes, methylene blue, thionin, etc. Basophil play important role in preventing blood clotting. The heparin inside the cell is a form of natural blood thinner that keeps the blood flowing through the body. Basophil is a non-phagocytic cell, less than 1% of total leukocyte. Basophil are the substance play role in a certain allergic responses. It is a two-lobe structure. Granule which is present in basophil are cytokines, histamine, and heparin. Next slide is about mast cell, also known as mastrocyte or leprocyte. It is a type of granulocyte derived from myeloid stem cell that is a part of immune and neuroimmune system. The cell produced in bone marrow in indifferentiated form but functional in tissue. Mast cell is located in the mucous membrane, and these cells also found in skin, connective tissue, mucosal epithelial tissue, digestive tract, and urinary tract. Functionally, it is similar to basophil cell, and marker is CD34. Here, CD means cluster of differentiation, is a protocol used for the identification and investigation of cell surface molecule. Now we discuss about monocyte. Monocyte is a leukocyte with bean-shaped or brain-like convoluted nuclei, circulate in blood with half-life of 8 hours. It is a precursor of tissue macrophage. Monocyte are the type of leukocyte or white blood cell. It is the largest type of leukocyte and differentiated into macrophage and myeloid lineage dendritic cell, also referred as a scavenger of body. Remove dead cell from the origination point. Marker CD64. It is a phagocytic cell, behave as an antigen presenting cell, APC. Monocyte, on the basis of location, different name has been given. If present in liver, known as Kupffer cell. If present in bone, known as osteoclast. If present in brain, known as microglial. If present in lungs, known as alveolar. If they present in kidney, known as mesangial. Now we discuss macrophage. Macrophage is a mononuclear phagocytic cell in tissue derived from blood monocyte. Participate in both innate as well as adaptive immunity. Macrophage phagocyte antigen and secrete proteins, monokines, that regulates cell involved in immune response. Macrophage are specialized cells involved in the detection and phagocytosis and destruction of bacteria and other harmful organisms. Macrophage can form bridge between innate as well as adaptive immune system. Next slide is about erythrocyte. Erythrocyte, also known as RBC, red cell, red blood corpuscle, hematids, erythroid cells. The red color of the RBC is due to the presence of hemoglobin. Hem means iron and globin means protein. It lacks cytoplasmic organelles such as ribosome, nucleus, and mitochondria. Composition is about 62% of water, 35% of hemoglobin, and 2.5% of sugar, lipid, carbohydrate, protein, enzyme, and ions. Function: various pigments are derived from hemoglobin after disintegration of RBC. It involves in acid-base balance and also maintain viscosity. Now platelets. 30% of the platelets are stored in spleen, released when required, and they are not actually cells but fragment of cells. Megakaryocyte derived in bone marrow play a role in clot contraction, and the granules which is present in platelets are microtubules, actin, myosin, filament, and they also play a role in inflammation. Now we discuss dendritic cell. Dendritic cell may come from lymphoid as well as myeloid origin. Marker is CD141, cell with dendriform star-shaped morphology. These cells secrete cytokines. Cytokines are a large group of protein, peptide, or a glycoprotein. They act as a messenger between innate and adaptive immune system, motile in nature, and phagocytic cell behave as an APC, antigen presenting cell. The main function is to process antigen material and presenting it on the cell surface to the T cell of the immune system. In this diagram, we can see that dendritic cell arises from both myeloid as well as lymphoid lineage. In this diagram, dendritic cell is present in skin, so it is called Langerhans cells, and the dendritic cell which involved in circulation known as migrating wheel cell. Depending upon location, different name has been assigned. If dendritic cell present in skin known as Langerhans cells, if present in liver, lungs, heart known as interstitial cells, if present in circulation known as wheel, and if present in lymph node known as interdigitating cell. Now we discuss lymphoid lineage, means the cell which arise from lymphoid lineage. In this, they are divided into two types: large lymphocyte and small lymphocyte. Large lymphocyte includes natural killer cell. Natural killer cell are present in blood and move into the other tissue to find targets. The immune cell can recognize and kills the cell of somebody's bodies that have been infected with a pathogen. Natural killer cell are the lymphocyte in the same family as T and B cell, coming from a common progenitors. It is a type of cytotoxic lymphocyte and also a major component of innate immune system, and play important role in the rejection of tumor and a cell infected by virus. The cell kills by releasing small cytoplasmic granules protein, which is perforin and granenzyme, that cause the target cell to die by apoptosis. Apoptosis is a form of programmed cell death that occurs in multinuclear organism. Biochemical events leads to characterized cell change and death. These cells include cell shrinkage, nuclear fragmentation, and chromatin condensation, and their marker are CD16 and CD56. Next we discuss small lymphocyte. It includes B cell. B cell are also known as B lymphocyte, are a type of white blood cell of the lymphocyte subtype. The main function of B cell are to make antibodies against antigen means they make particular antibody against particular antigen and to developed into memory B cell after activation by antigen interaction. There are two type of B cell, plasma cell and memory B cell. Plasma cell, once activated B cell may differentiate into plasma cell. Memory B cell, memory B cell will differentiate it into memory B cell when activated. B cell marker is CD19. Next slide is T cell. T cell, also known as T lymphocyte, is a type of leukocyte. T cell marker are CD3, CD4 and CD8 and also involved in adaptive immunity. There are type of T cell, that is cytotoxic T cell, helper T cell and regulatory T cell. Helper T cell have different co-receptor called CD4 on their cell surface, and regulatory T cell also have CD4 on their surface but do not activate the immune system like helper T cell. Regulatory T cells suppress the immune response in several ways, include producing anti-inflammatory cytokines that suppress the immune response. Thank you.